Welcome to the Nervous Breakdown podcast. Today we're going to be looking at antibiotics, how to target bacteria and cell structures alike with many different methods. So let's get into it. What is an antibiotic?
An antibiotic really is a chemical or some kind of protein that is interfering with the normal bacterial functioning. So a lot of the antibiotics target different processes and mechanisms that are utilized by bacteria for their survival. So let's talk about kind of in which way we're going to look at the antibiotics and how we're going to structure our treatments and where all these antibiotics come and where the interactions are with the bacteria.
Well, antibiotics act in many ways. They can inhibit the formation, synthesis of the cell wall, inhibit the inhibit mechanisms of protein synthesis by inhibiting ribosomes, translation, these kind of processes. They'll also prevent nucleotides from attaching onto it, preventing DNA replication and cell cycle as well. Will they be able to stop that?
So you can break down all the reactions that are occurring. All the reactions in nature come down into three classes. You have your class one, your universal reactions, which is really your energy production of your Krebs cycle, of your glycolysis, those kind of level of reactions, very universal in between species. Then you have your class two reactions, which is setting up your precursors. So these are your anabolic reactions, which you will use for then your class 3 reactions, and these are where you're forming large structures. So you're forming your polypeptides, you're forming your large structures within your cell itself. So there are different. There's different levels of selectivity in when you can target bacterial cells over human cells, because that's what you're trying to do here. Chemotherapy, as it comes down to it, is really using chemicals and different structures and compounds to try and target bacterial structures whilst being non-selective for your human cells, so selectively targeting the bacterial cells. So if we start off at the more basic level, we have a couple of drugs at the class 2 level. So this is coming into the anabolic reactions of creating your nucleotides. So this is in the folate pathway. So you will try to form folic acid, which is important in nucleotide synthesis. So drugs here are things such as your sulfonamides, so sulfamethoxazole. These inhibit your DHPS, an enzyme here, dihydroterate synthase, and yeah, so this is a key enzyme to convert your PABA into your folate. So these are PABA analogs that are able to competitively bind and inhibit this enzyme. Similarly, in the lower down stage of this reaction pathway, you have trimethoprim, which is a DHFR inhibitor. This can also be seen in things like cancer with methotrexate, which is also used for rheumatoid arthritis, to kind of deplete these highly proliferating cells of nucleotides. So in a similar way, it's used here as well. And what you can find with these drugs is because they're inhibiting on the same pathway, if you deliver them together, so combination therapy, this is really big in chemotherapy and especially for targeting in bacteria. If you're able to combine these two therapies, they are seen to have a much greater effect together rather than individually. So they are prescribed as crotremoxazole, which is trimethoprim, and sulfamethoxazole. Now we're going to look at some of the class 3 reactions. This is where your selectivity comes really into play. And these are some processes that are more specific to bacteria rather than humans. And at the top of this, we have cell wall synthesis. Now, why would you think that targeting the cell wall and the peptidoglycan pathway is beneficial and more selective at targeting bacterial cells rather than human cells?
So in bacteria, they have a specific cell wall made out of murin specifically. So
That's the glue that holds it together.
right, yeah, definitely. Cell wall, and this cell wall can be inhibited various ways using antibiotics. However, human cells do not have a cell wall, only a cell membrane. And Antibiotics will act specifically to the cell wall on bacteria. Remember, it has to bind specifically, so it will be complementary, as they say. And this idea of complementary is also seen, as we mentioned before, with Pablo sulfonamide, with very similar structures. So,
Yeah, so if we kind of take a look at the pathways that we're looking at here, so peptidoglycan cell wall synthesis is really happening outside of the cell. So you have your monomers, so your NAG and your NAM subunits. I'm not going to say what they are because I do not remember, but I think it's N-acetylglutamine is your NAG, and then, yep, you have your NAM as well. So these will be made inside the bacterial cell, and then they have to be transported out and added to this growing peptidoglycan chain, added into the matrix. And so, yeah, so you're building your cell wall outside of the cell where you really have low energy stores available there. So now let's break down this pathway. So first you have your NAG subunit. You add UTP and you form a UDP NAG. And then in the next step of this pathway, you form this UDP NAG to go to UDP NAM. You use enol pyruvate transferase. This is a target for an antibiotic called fosfomycin, which inhibits enol pyruvate transferase and kind of stops you being able to form your NAM subunit. The NAM subunit is really important because in the next step of the pathway, you're adding a short polypeptide chain. You're adding three amino acid residues, L-alanine, D-glu, MDAP or L-lysine here in this third position. And this is really important for linking the chains in between levels. It's a highly layered structure. So then in the next step of the pathway, you are adding in D-ala, the final two D-alas. And what you have here is you're able to use an antibiotic, cycloserine, which is a D-ala analog, very similar structure to D-alanine. But if you're able to add this in, then you're not able to form the peptide bond that will be important for bridging and adding the structure in between the layers of your peptidoglycan. So then once you're forming your UDP NAM with your 5 polypeptide chain, actually 6 polypeptide chain, no, 5, sorry, 5, you then are using the C55 carrier to get it outside of the cell membrane and get it to the cell wall. Now to do this, you form a pyrophosphate bridge with the carrier, and then a flippase enzyme flips the structure of this carrier to get this monomeric subunit out into the cell wall. Then here you have peptidoglycan transglycosylase to form the glycosidic bond between your NAG-NAM, which is one subunit, and add it to the growing peptidoglycan chain. And what you're able to do as well is you have your transpeptidase, which is forming the peptide bond between the MDAP or L-lysine of one NAM subunit and the fourth D-ala in another NAM subunit. So this is where you're able to form multiple layers of the peptidoglycan and really get a thick cell wall. Here is where you have the action of your beta lactams. Oh, sorry, I should mention that you also have bacitracin, which blocks the carrier and stops the dephosphorylation of the carrier to be able to reflip it back. So this process is just cycling through, pushing through your subunits to keep adding to your growing polymer. And so if you are able to block that with bacitracin, then you cannot keep outputting high numbers of monomers, high numbers of the subunits, so your cell wall really breaks down. Your beta-lactams block the transpeptidase enzyme. This is the real biggest class of antibiotic drugs, and they inhibit the cross-linking of your cell wall. So the different layers are not able to be cross-linked, and this makes the cell wall very weak, liable to breaking apart, loss of the structure. And what you get here is you'd get osmotic shock, ion imbalances, and the cell would swell and eventually rupture and burst. So your beta-lactams are in two forms, really. You have your penicillins and you have your cephalosporins. And you can manipulate the beta-lactams, and it's really taking the penicillins and altering their side chains to get the different desired effects that you want. So you can add a side chain, for example, to make it beta-lactamase resistant. Beta-lactamases, I must mention, are enzymes which are found in the periplasmic space, usually on gram-negative bacteria, and they function to cleave your beta-lactamase uh, inhibitors, your beta-lactams. They function to cleave your beta-lactams so that they cannot perform their function. It's really a resistance mechanism by the bacteria. So things like this are methicillin, which have a different side chain to your normal penicillin, your benzopenicillin, and so are beta-lactamase resistant. You can also add beta-lactamase inhibitors, such as clovulonic acid, um, and this functions to inhibit your beta-lactamase so that your beta-lactams can perform their function. You could also add side chains to make them more broad spectrum, and you have very broad spectrum antibiotics such as ampicillin and amoxicillin. You have your cephalosporins as well in a similar function, and you have carbapenems and monobactams. These are also beta-lactams, but they're more specific and they're mainly used as a last resort for gram-negative bacteria if your beta-lactams have really failed and your penicillins have failed. It's important to mention vancomycin, a very powerful antibiotic, and is often given for MRSA, methicillin-resistant Staphylococcus aureus, functions to form hydrogen bonds with the D-aller, with the fourth D-aller, the D-aller, a lot of D-allers there. Um, so it forms hydrogen bonds with the D-allers, and it doesn't allow for the transpeptidase to be able to form that bond between the MDAP or the L-lysine and the 4-D-alanine on the other NAGNAB subunits. So this way you cannot form that transpeptidase reaction, and you cannot form that peptide bond. So that was really looking into the cell wall synthesis pathway, one of the biggest pathways and the biggest targets for us, especially in antibiotic therapies.
I think we've gone through everything under the surface about antibiotics and over the surface, because we look at some patient cases, clinical treatment, all these things, prognosis, treatment, everything like that. Signs you look for. Hope you enjoyed this episode. Hope you learned a lot. Check out our other videos on the Nervous Breakdown podcast.